Okay, hi everybody. Thank you very much for coming along to the Perkins Open Day and also for coming along to my talk. I'm Grant Morahan. I'm the scientific head of the Clinical Sciences Division here at the Perkins Institute. I'm also head of the Centre for Diabetes Research at the Perkins Institute. So what I'm going to tell you today is some things that even your doctor doesn't know. I'll be telling you some secrets. We have some information that is leading the world, and it hasn't been published yet, so you're one of the first people in the world that's going to hear about these very exciting results. In my lab, we are tackling the world's biggest problems. These are the biggest health problems in the world. They affect literally hundreds of millions, if not billions of people around the world and cost trillions of dollars to deal with. So we're talking heart disease, cancer, diabetes, dementia. Now, most researchers will be very happy if they can make a big impact in any one of these diseases. Today, I'm going to show you how we can make a big impact in at least two, and maybe in question time I can show you three. One of the things that you won't hear is that even though we're living in an era in which medical technology is at unprecedented levels, and we're very lucky to be living in this time when the technology is so high, medical care has never been better. But even so, doctors can't tell you which people diagnosed with cancer are most likely to die of that cancer and who can survive. Which people with diabetes are going to go on to develop this very sad consequences of diabetes, like going blind, having kidney failure, having limbs amputated. 30% of people with diabetes have that fate. But we can't tell which people at the time of diagnosis. Doctors can't tell you which people, when they're diagnosed with high blood pressure, and about 30% of their population will have hypertension, can't tell you which ones of those people will go on to have a heart attack or a stroke. The two biggest risk diseases for high blood pressure is a risk for. Or which ladies, when they're pregnant, are going to have problems in childbirth that could kill them or their baby. So this is why we study genetics, because we think that these diseases have a genetic basis, and if we can find out the genes that cause these diseases, we'll be able to identify the people most at risk, and then we understand the disease better so we can develop new treatments that can be more effective in dealing with these diseases. And again, we're living in a very exciting time around the world. In the last 15 years, there's been literally thousands of disease genes that have been identified that are associated with all sorts of diseases. Now, here we're talking about complex genetic diseases. We're not talking about the simple diseases like Huntington's disease or thalassemia, where there's one mutation, usually a rare mutation, gives rise to that disease. Those are called simple Mendelian diseases. We're talking about complex diseases that are very common in the population, and these are caused by many genes that act together with environmental factors. So there's been thousands of these have been identified. It's been very successful, but most of these variants that have been found have very small effects. So their clinical significance is doubtful and they have little predictive value. So if you're diagnosed with a particular condition, your doctor's not going to ask for a genetic test to be done on you because with all these genetic markers so far, there's no clinical value in them. So I think we need a new approach, and this is what my group has been working towards over the best part of the last 10 years now. So the question we want to address is, can we identify people who are at high risk of a disease or a bad disease outcome that's caused by many genes, each having a small impact acting together? Now, how we're different from every other geneticist around the world. So if we, just to take a step back, we've got 3,000 million DNA bases in our genome, so 3,000 million letters in our genome. And if we look to see how many of those letters are variable between all of us individuals, there'll probably be between 1 million and 10 million. So it's a bit like looking up at the sky at night and seeing all the stars at night. That's just how many variable genetic elements that we all have. Now, the way that most geneticists around the world deal with this problem is that they look at one of these bases at a time, so they do a genome sequencing thing, And you may have heard a talk earlier today that this is how some rare mutations get found. Or they just study one at a time and try to find out how this has a role in disease. Well, this is not the best way to tackle the problem that I think. And I think that if you look at the stars at night, it's not useful to see one, but it's very useful when you can see patterns in the stars. So, for example, in the southern hemisphere, you can see the Southern Cross, and people have been using this information to be able to sail across the Pacific Ocean for hundreds of years. So, finding patterns in your genetics can be very important and can be just as useful as finding patterns in the stars. So this is the approach that we've taken, and that's very different from what everybody else around the world has been doing. So what we're talking about is studying 1 million genetic markers per person, and we're looking at experiments that have maybe 2,000 people in it. So we're looking at 2,000 million pieces of genetic information, and we want to see how each one of these can interact. So if you think about it, we have 2,000 million along one axis, and we want to see how they interact with another. That's 2 by 10 to the 9 by 2 by 10 to the 9, so it's 4 by 10 to the 18, which is a very, very big number, and we're just looking at 2 interactions. Now, we've done this, and many other groups around the world have tried to do this, but even looking in 2 dimensions, we can't see major interactions. So what we do is we have a very sophisticated mathematical and computational program where we don't look in 2 dimensions, but we look in 100 dimensions. And in 100-dimensional space, now we can start to resolve these interactions. So it's very difficult for humans to visualise or to think in 100-dimensional space. So this is why we have to use supercomputers to do this work. So I'll show you 2 examples. One is of heart disease. This is, of course, a major cause of death and disability in Australia. 40,000 people in Australia, unfortunately, die of heart disease a year, and over 1 million people are hospitalised every year in Australia. Most people know this, but most people are not aware that over half the people who die of a heart attack don't have any previous warning. So if we were able to identify people at high risk of a heart attack, they could get some education so that when they find the symptoms coming on, they could call an ambulance and that could save their lives. So we're trying to find a test to predict the risk of heart disease. Now, if you go along to the doctor to find out what your risk is of having a heart attack, they will ask you a few questions, take a blood sample, and send it off to a pathology lab to do some tests for cholesterol and other things. Then they come up with integrating all this information into a Framingham risk score. Now, in people with high blood pressure, that Framingham risk score is a predictive value of 66%. Now, you might think that's a pass, but if you think about it, tossing a coin is an accuracy of 50%. So it's not much better than tossing a coin. And this is the outcome in a study with 5,000 people in it. This and other graphs I'll show you are survival graphs. So we have the percentage of the population who are disease-free here, and we're followed up over time. So at 40 years of age, everyone is okay, but as time goes by, people in the population have a heart attack. This is the Framingham low risk, Framingham high risk, and really they're only significantly different by the time they're in their 70s. So you can't predict what's going to happen to someone in this range here, and even the prediction here is not that good. So how does our genetic test compare to this? So again, there's the Framingham risk score. How does our genetic test compare? Well, you can be the judge. The people that we say at low risk are really at low risk, and they have less incidence of heart disease than in the Framingham score. But look, you can tell here that people at genetic high risk, 70% of those have had a heart attack by the time they're 80. And there's a significant difference between the 2 groups here in their 60s. So we have the technology that can identify people who are at high risk of having a heart attack. And so that knowledge could well save their lives. And our test has an accuracy of 92%. So we did well against the standard test. What about the gold standard test? Now, the gold standard test in the world is called cardiac computed tomography. angiography. This is a big mouthful. What it means is that they take a CAT scan of your heart. So using this very sophisticated computer and X-ray machines, they can see high-resolution images within your heart. And this is useful for people who have angina and predicting their outcomes. So we tested people that were low risk by CCTA, as defined by the world's leading cardiologist in Scotland. So we looked at the people who they said had a low risk of having a heart disease based upon this CAT scan of the heart. And we found that the high genetic risk cases were more likely to have heart attacks and more likely to die of their heart attack. So this is some medical terminology. So these people who had low attenuation plaque but high genetic risk were 18 times more likely to have a heart attack than people at low genetic risk. And low calcium score, this is another thing that cardiologists find very useful. People with low calcium score but high genetic risk were 5 times more likely to have a heart attack than people with the low genetic risk. So our genetic test is better than the gold standard test run by the world's leading experts at that technology. So what about melanoma? So as you all know, it's the most deadly of the skin cancers. Australia is the world capital of melanoma, and it's the third most common cancer in Australia, affecting 15,000 people a year. So we've developed genetic tests that can predict someone's risk of dying of melanoma within 10 years of diagnosis. Again, let's look at how the tests perform. The gold standard medical test here, the gold standard medical test here is the Breslow test. What happens here is that the melanoma is taken off and it's examined under a microscope by 2 highly trained doctors, a specialist in tissue examination, and they can say whether the person is low risk or high risk. So 20% death rate in the low risk people over 10 years and 60% in the high risk people. Here's our genetic test. Genetic low risk people, less than 4% deaths. Our genetic medium risk group is doing just as well, or probably a little bit better than the Breslow low. But our genetic high-risk people, we can identify people, 90% of these people, unfortunately, will die of melanoma within 10 years. Breslow test is no good at identifying risk beyond 5 years. So our test beats Breslow, it's more accurate, and it can predict survival over 10 years. Again, here's how we compare with the gold standard. The Breslow test has a relative risk of 4. That is, someone told by their doctor they're at high risk is 4 times more likely to die than someone who's seen as low risk by that test. Our genetic test has a relative risk of 15. So the people at high genetic risk are 15 times more likely not to survive. So why does this matter? It matters because people at low risk by the Breslow test don't get any treatment, and yet you can see that 50% of the genetic high-risk people will die within 10 years. Those people are told they're at low risk and told to go away and only come back if their melanoma comes back. And by the time that happens, it's too late. On the other hand, people told by their doctor they're at very high risk of disease, if they have the genetic low risk, are very likely to survive over 10 years. Let's look at this again. People with stage 1 or 2 melanoma don't get any treatment. As I said, they are told to come back if their melanoma comes back. We can identify those people at high risk, even with early stage melanoma, who are likely to die. So these people should be getting treatment instead of waiting until their melanoma comes back. And on the other hand, we can find survivors even with stage 3 melanoma. So 100 years ago, these people would have said they prayed to their patron saint or whatever and survived, so it was a miracle. It's the miracle, all right, but it's a miracle caused by their own genes that's doing this. What about predicting whether someone can get a melanoma? And now so prevention is worth a pound of cure. Can we identify people who could develop melanoma? Now, looking at traditional risk factors, the relative risk is very low. So even though you say people with red hair or have childhood sunburn are at risk, it's not really very predictive. Here's our test for people who have moles on their skin and who had childhood sunburn. So we have now a relative risk of 14. These people with high genetic risk are 14 times more likely to develop melanoma than someone with a low genetic risk. So we can identify children, for example, who have a particular genetic risk, and those children can be more alert about exposure to the sun and in later life getting their skin checked. Here is another genetic risk test. This is for people who don't have any traditional risk factor. So this is for people with dark hair, and dark hair is not considered a risk factor for melanoma at all. And yet, have a look at this. If they're genetic low risk, none of these people got melanoma. If they're high genetic risk, they all did. The relative risk is 93. This is just an extraordinary result, and it shows you the power of our technology. We can identify people most at risk of disease or most at risk of the worst outcomes of those disease, and we can do this just on testing their DNA from a saliva sample. We don't need a blood sample even. So the benefits, our genetic tests allow earlier diagnosis and better management of the patients, and potentially in the future we can develop new treatments that are based upon the actual DNA markers, not based upon some other things that don't work. So if we were in America, there would be 100 people on this slide here, but most of the work was done by myself and Sylvia, postdoc in my lab, and for the heart disease test, we worked for a new professor of cardiology at Perkins South, and collaborations with David Newby, who leads the leading team in CCTA in the world, in Scotland. So no one can tell where the future could lead to. We now can tell where the future may lead in terms of health risks. We can do this from our genetic tests. So your healthy future is like a long road ahead, and if there was going to be an obstacle, a dangerous obstacle in that road ahead, wouldn't you want to know now so you can plan ahead and avoid that obstacle? We can now do that with our technology. So we think that your DNA holds the secrets to your future health. And here at the Perkins, we're working to find out what those secrets are so we can bring you a healthier future. And thank you very much.
Could I ask, the genetic markers that you identify, are they particular to the Australian community, or have they been developed on an international basis?
It's a good question. Most of these tests have been developed on international cohorts because it's too expensive to do these tests in Australia. So we've developed the heart disease test on Europeans, the test for melanoma is with American patients. But where we can look at these tests in Australia, we do find out that they also confer risk in Australia.
Grant, do you use the supercomputer at Curtin?
We started off using that, but we've now refined the method so much that we can run it on a desktop computer. So we're doing it all now within Perkins. Yeah, it took more than a week on the supercomputer, but now it takes a week on our desktop. So while people are thinking about this sort of thing is called precision medicine, and everyone knows that precision medicine is the future, but nobody in the world can deliver precision medicine for these common genetic diseases that we've been talking about today. Yes.
I guess for more with the heart disease, with the high-risk genetic people, have they looked at has there been any looking at data for those who are leading like your traditional sort of healthy lifestyle versus that, and how that correlates if you are in that genetic high-risk situation?
That's a very good question, and here we have that result. So these people were part of a big study where they were followed up for over 20 years, and they had all sorts of medical examinations. So this summarises the information from this study. So we have men and women, low genetic risk, high genetic risk. Here's their cholesterol, HDL levels, blood pressure, average blood pressure, whether they were treated for their blood pressure, whether they smoked. There's no differences between any of these things between these two groups. What's giving these people heart attacks is their genetics. That's not whether they're smoking. That's not whether they're getting blood pressure treatment. That's not protecting them from heart attack. It's not their HDL, it's not their cholesterol. So you could have low cholesterol, not be a smoker, do everything right, but if you've got high genetic risk, then you're at risk. So we really need to develop new treatments that are based upon the genetic mechanisms that we're uncovering, because the existing treatments are not saving those people.
So all those years of public health campaigns might have been taxpayers' money not best spent.
Yes. Taxpayers' money would be much better spent in my lab.
Have you noticed anything that will mitigate the effects of the genetics on the risk of heart attack?
No, because we have to use these data that have been collected over time, and we're not doing anything prospectively. That would have to be done via collaborations. Yeah.
I just observed the slow onset of questions, because I think we're all quite overwhelmed with what you've presented to us.
Thank you.
And just absorbing it. It's an amazing amount of information and significance of information. How far off is having this saliva test?
In theory, we could do it today. In practice, we have to wait till we get government approval to sell those tests. So that will cost hundreds of thousands of dollars and take 1 or 2 years. So that's unfortunate, but that's just the way it goes.
And then for the examples you gave with the heart disease and melanoma, for example, one salivary sample would cover that whole scope of risk?
In theory, yes, it would. So one saliva sample we can test, in theory, we can test 1 million genetic markers. And so we can get all this information out.
This is just mind-blowingly.
It is, and it's even more mind-blowing if I tell you that.
Congratulations.
It's even more mind-blowing. We could do this test on a baby and predict that baby's risk of having a heart attack in 70 years' time, or developing melanoma in 60 years' time. This is the real future of medicine.
Is the genetic markers going to be different for each form of cancer, or how will that work?
Yes, it probably will be. It's quite likely it will be, but we need to test that out. So it's likely that what's involved in melanoma is an immune response. I think it's going to be a battle between the biology of the tumour and the host biology. What I didn't tell you is all these genetic markers are from the person. They're not genetic markers of the tumour. So again, a lot of people look at the mutations in the tumour. We think that's not really relevant. What's protecting those people is their own DNA. And so that's probably pointing to them making a good environment or a bad environment for their tumour to grow in. So we want to find out what makes that bad environment so we can provide that information to people who could then use that to save their life.
Sorry, I just want to ask another question on the basis of that. So then your genetic inheritance becomes increasingly important to you. Choosing your parents becomes increasingly important to you.
That's true for simple Mendelian diseases. Remember at the start I talked about simple genetic diseases where one gene causes the disease. Here we're talking about interactions of maybe 10 genes or more. And so when you get to that level, and these genes are also very common in the population, whereas the rare, simple genetic diseases, those mutants are rare in the population. So when you have 10 genes that are very common in the population, its family histories really have very little influence in it. And again, family history is not predictive in the heart attack, and it's not predictive in melanoma development in our cohorts either. So if anyone has a family history, that you need to be tested yourself, not worry about so much about what else is happening in your family.
It seems that Western Australia is the leader in this technology in the world. Do we have a database, or a genetic database from which we can work and pursue not only the heart attack and melanoma, but the whole range? Because not that I know much about it, but we used to have the Busselton survey, which was the epidemiological treasure house of the world. Yes. We should be in a position to develop a database, a genetic database, to pursue all these things that you talked about.
That would be wonderful if we could do that.
But what does it need?
Well, the information is kept there by the health department. I don't want to tell stories out of school, but for us to get that information out of that health department, it takes us at least 2 years to get that, and it costs $60,000 to $100,000. And in today's world, no one's going to pay a researcher $60,000 to do work 2 years in the future. It's just not going to happen. So West Australia, yes, it does have all this epidemiology and all these public health records, but they're just not accessible to researchers, unfortunately.
What about using the 23andMe and Ancestry databases? Because 23andMe in particular tests for several genetic markers.
So 23andMe are Americans. Americans have got an NIH policy that's not invented here. So they think if it's not invented in America, it can't be any good. We've approached 23andMe, they just said, go away, we're too small.
What about because they're working with the federal police to give them access to the DNA profiles? Would that create a pathway for you guys?
Yeah, so if people have had their genotypes done by 23andMe, then it's possible for that information to be used in our tests.
So we should just do 23andMe and then send you the results? Would that work?
That's possible, yes. But
We're going to have to wrap it up. Thank you very much, Grant.
Well, thanks everybody for all your questions.